So, hey everybody, welcome to the first of our special topics lectures. It is my pleasure to introduce Dr. Joel Wertheim. He's a professor in the School of Medicine, right? You're in School of Medicine, right? Yeah. He's in the School of Medicine. His research area focuses on viral molecular epidemiology. He's done a lot of work with the CDC behind HIV molecular surveillance, so kind of tracking how HIV spreads across the country. He and I have also done a lot of work related to COVID origins and just kind of studying COVID epidemiology, particularly throughout San Diego County. So yeah, hopefully his lecture is going to be super relevant to stuff you all have not only learned about but lived through over the past few years. But yeah, let's welcome our guest. Thank you, Joel. Thanks for being with us.
Right. Thanks for having me. If some of you wouldn't mind turning your cameras on, it's always nice to be able to see some faces of the people I'm talking to on Zoom. So if you're willing to do that, uh, please click that button now and get started. Hi. So as Nima mentioned, I'm Professor Joel Wertheim. I am professor in the School of Medicine here at UC San Diego. I am trained as an evolutionary biologist, so I'm not a real doctor. I study the evolution of uh, sort of the molecular evolution, genes and genomes, specifically of viruses. For example, today we're going to talk about a virus like this one, SARS-CoV-2, which everyone is intimately familiar with, but we'll also touch on a couple other viruses. And the topic that I want to talk to you about today is one that's particularly close to my interest and lifelong passion, which is the origin of viral pandemics, a topic that used to be a bit more esoteric and now is, of course, a lot more relevant to all of us. And I'd like to start by introducing you to this gentleman here, one of the most famous scientists whose face you don't recognize. Um, so, does anyone have any idea who this guy is? And that's okay because I just lectured to a group of juniors and seniors here at the university, and not a single one of them could pick him out either. But I promise you, you've heard of this guy. So his name is Linus Pauling, or at least you've heard of his legacy. He is famous for being the only person to win two Nobel Prizes where one of them wasn't in science. Of course, Albert Einstein and Marie Curie also won two Nobel Prizes, so nothing too special there. But Pauling was famous because one of his prizes was a Nobel Peace Prize, and the other one was in chemistry. His first prize was on how atoms bond to each other. So you may have come across the term Pauling bonds in a chemistry class. That's Linus Pauling. And of course, he was also famous for his work for nuclear anti-proliferation in the 1960s, 70s. So both still very relevant topics. But as sort of famous and successful as Linus Pauling was, he did not get everything right. For example, he was famous for advocating that DNA, the molecule that contains the information of life, was a triple helix, which was disproven in the early 1950s, showing that in fact it was a double helix. Unfortunately, Linus Pauling was a good enough scientist to, after Watson and Crick and Franklin came out with their crystallography evidence showing that DNA was in fact a double helix, he abandoned his idea that the molecule of life was a triple helix. So good for him there. Unfortunately, he also had other ideas, specifically that vitamin C could macrodosing of vitamin C could cure basically every human ailment, specifically boosting the immune system, which is not supported by scientific evidence, but he stuck with this one until the day he died. So if you've ever been offered a glass of orange juice to cure a cold, or been told to take Airborne, the vitamin C supplement developed by a school teacher, you have Linus Pauling and his death grip on pseudoscience to thank for that. So, brilliant guy, Not all of his ideas were right. Some were very wrong. But he also came up with a concept that's key to molecular biology and one that I use in my day to day research. And that is called the molecular clock, which is something that you may have come across in a biology class, but may anyone, just show of hands, anyone come across this term in biology so far? This is something you'll encounter in university then. I don't remember learning about it in high school either, but it is definitely my favorite concept in all of molecular. And what it is basically the steady accumulation of mutations over time. And the way we can think about this,
Like a mutation.
Cordyceps mutated.
Was some sort of mutation.
Mutated them into horrific monsters!
A mutation.
Why? Because we are mutants.
The pangolin has mutated how?
Any mutations, augmentations?
Mutated the dinosaur genetic code.
They are mutated, Seabass.
Are mutations caused by radiation?
All through mutation.
That better not be the mutants.
is a lot of mutation are constantly happening everywhere, and eventually they fix in the population. And you can see this here in a toy example. I've picked up a virus. This virus only has 50 nucleotides or nucleic acids in its genome, A's, T's, G's, and C's here. And you can see that in year 1. Let's zoom in a bit. We can see here's our sequence of A's, T's, G's, and then after about a year, we have a single mutation going from G to A. This is a substitution mutation. And then in year 3, that virus mutates again, and now we keep that old G to A, and now we have a C to T mutation. And then in year 4 and in year 5, we can see we accumulate more and more mutations all the way up to year 6. So now we have 6 mutations, about 1 mutation in this genome per year. And that steady accumulation of mutations is known as the molecular clock. And it allows us to say if we have one, and if we know that rate of mutations over time, we can say we have a sequence in 1 year and a sequence in another. They must differ by 6 years of evolution going forward in time. And viruses, in fact, do evolve this quickly, and we can, in fact, look at them evolve week to week, month to month, year to year.
Some kind of mutation.
A lot of mutations. Okay, so the first virus I want to talk to you today about is influenza virus, which has been circulated, which we've been tracking its evolution for well over a century now. Here we have what's a phylogenetic tree showing the relationship between these viruses, and I'm going to zoom in here so you can see a little clearer. So that sort of ladder-like structure there, those are viruses, influenza viruses that were sampled between 1933 at the bottom and 1987 at the top. And you can see the year of sampling plotted on the x-axis and the number of nucleotide substitutions separating each of those sampled viruses from the base or the root of the tree on the y-axis. You can see that there's sort of this nice linear relationship between them. With the ones sampled in the 1930s and 40s, closer to the root of the tree and closer to the left and part of the x-axis. And as you move up into the tree, you move forward in time and you get more mutations or nucleotide substitutions away from them. And we can actually see this happening in time. So if we were to go back in the 1930s and just look at the genomic diversity of this virus, we can see that on average, we're only about 10 to 20 mutations from the root, which would make sense because the root of this tree goes all the way back to around 1918 and the great H1N1 Spanish influenza pandemic. And as we go forward in time, you can see now into the 40s through 1950, we start to accumulate substitutions, and we sort of move up this regression line. And if we continue to do that through the 50s and into the 1960s and into the 70s, you can see that we're gradually accumulating mutations over time, and viruses that we sampled before are the ancestor or close to the ancestor of viruses from the previous day. And we can see that through the 1970s and now in the 1980s, we just keep accumulating those mutations. That steady tick of nucleotide substitutions over time is the molecular clock. So we have a pretty good idea. If the only thing you knew was the number of nucleotides that this virus were separated from the root, you could guess its year of isolation pretty closely. And that's going to become a very powerful tool. So now the nice clean example is with from influenza. Now we're going to switch over to HIV, or the human immunodeficiency virus, a virus that before COVID was my main topic of study, and now post-COVID is again my main topic. And I'd like to talk to you about how the HIV-1 pandemic began, specifically when it started, where it started, and the first thing we're going to address, how it started. But in order to understand that, we're going to have to actually learn how to interpret these phylogenetic trees that I just showed. Okay, so let's start here. This is a phylogenetic tree. When I don't study viruses, my passion is primates. So I always like to use primates as an example here. You will recognize some of these organisms. We have chimpanzees, and we have humans, bonobos, and gorillas. We'll start here with humans, and we'll use the example here of Charles Darwin, shown in his pre-bearded phase. It's just a reminder to all of you young folks that, in fact, the old bearded scientists of the 19th and 20th century were also, in fact, at one time, young people too, doing their science. It's important to remember that, Darwin did all of his great work before he got a beard. Einstein did all of his great work before he got crazy hair. And Marie Curie died young. So young people do great science. Good to remember. Okay, so in our phylogenetic tree, we have two things. One, we have nodes, in this case represented by the tips on the tree. We have humans and chimpanzees and bonobos and gorillas. These are the African great apes, humans. And then we also have on our tree, we have branches, and those represent evolutionary relationships that we don't observe in present day, that we have to infer into the evolutionary past. And using these branches, you can see that we have branches that lead to the tips of the tree. And then we also have internal branches that represent the relationships of these species within the middle of the tree. So we can use a phylogenetic tree to ask the question, who is our closest living relative? Ours being humans. And to do that, we have to identify a common ancestor. And this is the most important thing about reading a phylogenetic tree, is you have to read it going back in time. So we start here with humans because we're saying who is the closest living ancestor to humans. And we can travel back in evolutionary time until we hit sort of an ancestral node here in the tree. And then we sort of turn around and we go forward in time. Oh, sorry, first. So this is the point in evolutionary time where humans share a most recent common ancestor with someone or something. And as we go forward in time, we can see that we don't encounter just one species, but in fact, as we move forward in the tree, we not only encounter chimpanzees, but we also encounter, in the same evolutionary distance, bonobos. Means that humans don't have a single closest relative. We are equally closely related to both chimpanzees and to bonobos because we share a common ancestor with chimpanzees, that star on the phylogeny, at the same point in the tree with both. And all three of us share a common ancestor in that tree before any of us get to the deep branch that connects us to gorillas. Now, what this means is that chimpanzees and bonobos are more closely related to each other than either of them are to humans. And that's how you can sort of infer where is this most who shares a common ancestor. Oh, there we go. I had the star for chimpanzees and bonobos line. So they're more closely related to each other. Okay. But how do we know this? Now, I just showed you that tree, and you took it at face value, and you didn't question me at all on it. Well, we know this because of DNA, specifically the relationship between humans, chimpanzees, and gorillas was first worked out by sequencing the mitochondrial genomes of all three species, and that was done almost 30 years ago now. And that's how we first. Before that, there was endless scientific debate about whether humans are more closely related to gorillas or whether gorillas are more closely related to chimpanzees. And our good friend DNA here sorted it out for us. Just lets you know that usually when there's a discrepancy between the fossil record and the genomics are right. Okay. So that brings us back to HIV. Now, the HIV genome is much, much, much smaller than the human genome. Humans, we have about 3 billion nucleotides comprising our genome. Here, let's just zoom in here on the HIV genome. You can see it all there. That's about 9,800 A's, T's, G's, and C's. That's the whole shebang. The entire virus, its entire genome is on your screen right now. And assuming you have a good internet connection, it is visible and decipherable to you. This is the pathogen that's gone on to infect nearly 40 million people worldwide. We are no closer curing it today than we were 20. And in fact, the part of the genome that I look at in my day-to-day research is actually only about this big. So Nima and I work together, tracking HIV spread around the country on behalf of this disease control and prevention. And we just look at this tiny short fragment here, only about 1,497 new. And that's sort of what's used. And that's sort of the practical part of the genome. But today, we're going to focus on the whole thing. And I'll show it to you here. This is actually what it looks like in color. You can see that it's a pretty diverse virus. A lot of every time that color changes, those are different nucleotides. And we can use the HIV genome sequences to ask when and where did it start? And we can ask, how did the HIV pandemic begin? And let's go to a couple of competing hypotheses. But in order to address those hypotheses, or what we really want to know is when and where did the most recent common when did this most recent common ancestor of HIV exist? And now HIV1 group M is shown here on this phylogeny. Rather than showing it in a rectangle here, it's displayed on a circle. You can see these sort of different subtypes that are common around the world. B is very common in the United States and North America, the rest of North America and Europe, and in parts of East Asia, whereas C and A are much more common in sub-Saharan Africa. But you can see that the most recent common ancestor is sort of at the root of the tree, and that's when we want to know, when did that virus, the most recent common ancestor of all of HIV, exist? And we will now examine 3 hypotheses for how it. The first, and one of the most pernicious, is that the CIA created it in a lab. The CIA was founded in 1947 after World War II. And when I say that, has anyone heard this as sort of a general conspiracy theory out there? Because it was very popular for a very long period of time, and is still popular in certain segments of the population. And there's a reason for it. And I'll show it to you here. And we're going to go back to something called Operation Infection, which, if you've never heard of this, I would say go Google this when we're done here today. Spend 20 to 40 minutes watching this New York Times documentary because it's a great display of how the world is actually working. And it's terrifying. And here we're going to see an example that says it's actually been like that for a very long time. Okay. So, why do we think that HIV was created by the CIA in a lab? And that's because here in 1984, I was alive for this. An article appeared in an Indian newspaper called The Patriot, suggesting that AIDS was the result of Pentagon experiments to develop a new and dangerous biological weapon. This may sound like origins of another virus. But this appeared in an Indian newspaper and eventually sort of took hold. Now, the question is, what's the story behind this story? And I'll let you listen to it.
Okay, so to start, let's go back to July 1983 and all the way over here, New Delhi, India. This is when a remarkable story appears in a newspaper called The Patriot. It claims the HIV virus was secretly created by US government scientists as a weapon to kill African Americans and gay people. It even names a facility, Fort Detrick in Maryland, where the virus was supposed to have been concocted. It's a crazy allegation printed in a small newspaper. No big deal, right? But fast forward just a couple of years and look what's happening. The story is spreading all over Africa. A scientific report's even published by 2 East German biologists who say they can prove AIDS is made in the USA. All these articles are from just a few months at the end of 1986. And then somehow it ends up here.
A Soviet military publication claims the virus that causes AIDS leaked from a U.S. Army laboratory conducting experiments in biological warfare.
Fairly shocking. I understand I'm talking to a younger generation here, so I need to explain something. First of all, that guy is Dan Rather. He was a very famous newscaster, one of the most famous people in the country. His job was to read the news on TV every evening for all of America. Let me take a step back. The evening news was something that aired every night on TV. Again, television was something that we used to watch and we couldn't control when it was on, and it just came on at 5:30 or 6:30 and we had no choice. Okay, we're all caught up now. Okay, so what's the story behind that Patriot News article and how did it make it onto what was legitimately one of the most respected news sources in the country?
This is just the perfect example of a very effective disinformation campaign.
Well, almost perfect. There are some obvious grammar mistakes here which tip off experts like Kathleen. Like in English, we'd say flu virus, not the virus flu.
It's written by a non-native English speaker, and it probably was written by a Russian language speaker.
They said, oh, the Indian newspaper, The Patriot, which we knew the KGB used this as an English language newspaper as a way to get stories out.
This was a classic Soviet tactic. Oleg Poluchin's another ex-KGB agent we found. He told us they'd always try and place the story
in a third world country,
somewhere like
in India, Thailand,
where journalists could be easily tricked
Japan,
or bribed.
That gave the story acceptability when nobody was searching about the origin.
The KGB let the story go quiet for a couple of years after India, but with AIDS still making scary headlines in '85, they revived it, this time in a prominent Moscow newspaper. And the source for this story? You guessed it. It's brilliant, really. They've repeated the story, but concealed their hand, distancing themselves from the lie they started.
So, disinformation is nothing new. And I think we can, since we know sort of from soup to nuts how that story started, we can say no. In fact, the CIA probably did not create in a lab, and it was just a KGB Soviet propaganda or disinformation. Okay, so the next possible theory for the origin of HIV involves the development of the oral polio vaccine, which we're not talking about the Salk or Sabin vaccines. Here we're talking about one that was developed by another scientist named Hillary Koprowski, who was working in what's now the Democratic Republic of the Congo and then Zaire in 1957 and 1960, trying to develop a polio vaccine. And he was presumably using Roundup primate tissue, which we know could be the source of viruses. We know that there was something called a virus called SV40 that was found in samples that were being used to develop this type of vaccine. So it is a theoretical possibility. And now, the oral polio vaccine, although it became prominent in the scientific literature, actually got its start in a news magazine called Rolling Stone, which is still published here. This is clearly from 1992, and you can see on it, after Wayne's World and Elton John, we have The Origin of AIDS, a New Investigation. Now, this article spurred a reporter named Ed Hooper to go and investigate it further, and he ended up writing a book called The River, which argued that during the development of a failed oral polio vaccine in the DRC, they were taking chimpanzee carcasses that they had hunted, grinding up their kidneys to grow the polio virus to be killed and then administered as a vaccine, and suggested that within those samples, SIV, or the simian immunodeficiency virus from chimpanzees, was transmitted to humans and gave rise to the HIV epidemic. Now, what's important here is that this scientist, Hillary Koprowski, was doing his work in the eastern part of the country, in Kisangani, far away from the epicenter of the early AIDS pandemic in Kinshasa. And also, I'm just going to put this little star here on the map for no particular reason that may or not be relevant 5 or 6 slides. So just make a note of where. Okay. So how do we know that the oral polio theory isn't correct? Now, keep in mind that the oral polio work was being done in the late 1950s and 1960s. So back almost 20 years ago now, a group of scientists using a massive supercomputer at Los Alamos National Laboratory, where a lot of the early HIV work was done, because that was the only place in the country that had computers sophisticated enough to carry it out. Of course, 20 years ago, nowadays, you could on your laptop, probably while streaming this. So lot of computational advantage. But they took all of the HIV sequences that were available at the time, and they plotted them out on based on the year of sampling and the branch length, or the number of substitutions from the root of the tree. And they basically drew this regression line. Very similarly to what we did for H1N1 influenza. However, for HIV, we only had samples going back to the early 1980s, 2000. So you had to extrapolate much deeper in time, which gives you a lot of variance in that age estimate. But they estimated that the most recent common ancestor of HIV circulated sometime in the 1930s, anywhere between, say, 1940 and 19 shown here on this graph, which seems pretty reasonable and would reject the oral polio vaccine origin hypothesis, because HIV would have been circulating in humans for decades before the oral polio vaccine. And in fact, they they went one better. Rather than just looking at contemporary strains, they also took a virus that had been pulled out of a freezer that had been stored since 1959, long before we even knew that HIV was a thing. And some people got a very, very short fragment of that 1959 sequence. And you can see it here on the tree. Now, what's important is that this 1959 sequence looks recessed compared to all of the other HIV-1 samples, from the subtype B to the subtype D to subtype C, etc. The 1959 sequence looks deeper into the tree, and that's because it was sampled decades earlier. And what's remarkable is that when you then take that 1959 sequence, take the length of its branch to the root of the tree, and you say, here's where we know that branch length should be, that ZR59 branch length shown here on that horizontal line, and you say, based on our molecular clock regression, what year do we think this virus would have been sampled? It looks like a virus from 1957, which is exceptionally close for for sort of a deeply sampled virus, and you can see with all of the sort of variants and when the ancestor would have occurred, really suggests that not only is that virus from 1959 likely an actual sort of old HIV sample, but also that it supports that HIV has been circulating since the early part of the century in humans. Means we can cross off the oral polio vaccine. That activity, while potentially risky, happened well after HIV was already circulating and diversifying in humans. So it wasn't the CIA, and it wasn't a failed vaccine experiment. So that leaves us with the, I guess, obvious answer that HIV was the result of a zoonotic jump of a virus from chimpanzees. Are known to have, as I mentioned, their own version of this. It's called SIV, or simian immunodeficiency virus. And in fact, we have sampled it, as scientists, from chimpanzees all over sub-Saharan Africa. And if you look here, you can see that the human virus had HIV-1, M, A, and B. That's the pandemic lineage that I've shown here and have the black arrow pointing at. You can see that it shares a most recent common ancestor with chimpanzee viruses in red there. Now we have chimpanzee viruses all over this tree, and in fact there have been many jumps of HIV into humans. HIV-O at the bottom is a separate introduction. HIV-N is a separate introduction into humans from chimpanzees. But HIV-1 group M is most closely related, or shares the most recent common ancestor with a population of chimpanzees in southeastern Cameroon, shown there in red. So this is the exact source population for the virus that went on to cause the HIV pandemic, and it's very close to Kinshasa, but it's not in Kinshasa. So the virus jumped from chimps likely into a hunter who, while butchering a chimpanzee, likely got blood on cut. It's called the cut hunter hypothesis. So you would have blood-to-blood contact, which is an ideal route for HIV transmission. Probably wasn't through the consumption of meat. So people in this region, time immemorial, have been hunting, butchering, and eating chimpanzees, but cooking would likely kill the virus. It was probably during the butchering process that this virus made this. But of course, this happened well over 100 years ago, and we have no direct evidence of that. But we do have phylogenetic inference that is strongly consistent. Okay, that's the origin of the HIV-1. What about our more recent pandemic, COVID-19? How do we think that one got started? It's a good question. I think we'll answer it now.
Oh.
Okay. Sorry, do you have a question? Happy to pause for questions. If you put them in the chat, I can see that, or take them. Okay, so I'm a scientist working in the United States. I want to know how COVID started. What's a good jackbooted government scientist to do these? Oh, with the government thing. You just go to www.covid.gov and you're greeted with this website. So clearly, the government is of strong opinion on how the pandemic started. Now, I could spend 1 to 2 to 3 hours walking you through every single point of misinformation that is on this official government website. But I think I've already established to you that sometimes governments mislead or are misled for their own purposes. So we're just going to take that as a given. And we're going to concentrate on the alternative hypothesis here. And that is the Huanan wholesale seafood market in Wuhan, a city of over 10 million people in China, shown here. Now, one thing that you may have heard over the course of the pandemic was that the WHO, the World Health Organization, went to investigate the Huanan seafood market as the possible location of origin, and they concluded here, this is from their report from 2021, that there were no verified reports of live mammals being sold at the market in 2019. Of course, live mammals are what you would naively expect to be the source of the SARS-CoV-2 pandemic. That animals like palm civets and raccoon dogs were the source of the SARS-1 outbreak or epidemic in 2022 through 2024 through the sale of these animals for fur and meat consumption. So potentially that's how this other related coronavirus got into humans. But the WHO said there were no animals being sold at this market in Wuhan, except there was this group of scientists, both Chinese and American, who had been tracking tick viruses in animals for sale in Wuhan in 2019. And after the WHO published their investigation in 2021, this group published their paper, and in it, they detailed animals that were for sale at the Huanan wholesale seafood market in Wuhan. And you can see here that we have snakes, we have hedgehogs, and in part D there, if you zoom in, you have this really adorable looking animal that's it looks like a raccoon, but in fact it's very closely related to a fox, and that is a raccoon dog, which is the same animal species that was known to be infected and transmit SARS during the SARS epidemic. So it's us, says my kid. Okay. So then we started writing a bunch of papers about this, and here's just one article says, group of scientists presses a case against the lab leak theory, which was sort of our first foray in fighting disinformation. And we ended up publishing several papers on this topic, one of which I'm going to highlight here, called The Molecular Epidemiology of Multiple Zoonotic Origins of SARS-CoV-2, which I co-authored with your professor or teacher here, Nima. So just this was not the first piece of work Nima and I had ever done together, but potentially our most impactful. Okay. And in that, we went to go build a SARS-CoV-2 phylogeny, looking at the genomic diversity of this virus in the earliest weeks and months of the pandemic. And you can see here we built this tree. It is not very diverse. Sort of every section on that branch is a single mutation. And I'll just show you here that back. In the beginning of the COVID-19 pandemic, this is before Alpha and Beta and Delta and Omicron, there were just two lineages. There was lineage A and there was lineage B. And you see about 2/3 of the tree is lineage B and about 1/3 of the tree is lineage. Now, right now, I'm going to give you a crash course in everything you need to know about the early genomics of SARS-CoV-2. And as first, what to know about lineage B. Lineage A, it differs by lineage A by just 2 mutations, and lineage B is associated with the earliest case of COVID-19 from the 10th of December, 2019. And in fact, every human case at the Huanan seafood market had the lineage B virus, and we found the lineage B virus in environmental samples taken at the Huanan seafood market as well. Now, in contrast, lineage A wasn't seen until late December in 2019. In fact, the first cluster of human cases, a mother, father, and their adult son, had the lineage A virus. Now, they lived near the Huanan seafood market, but didn't actually go inside and didn't have any epidemiological connection to the market itself. So as I said, there also another early lineage A case was staying at a hotel near the market, but again didn't have any direct connection to. Now, what's important to know is that the lineage A virus, those 2 mutations that separate A and B, A is actually 2 mutations closer to the bat ancestor than lineage B. I'm showing here that they're only about 5 or 6 mutations away. In fact, they are hundreds of mutations away. But along the way from bat virus to lineage B, you need to walk through the lineage A genotype. So both of those mutations put A closer to. So we need to turn to our close friend, the molecular clock here, to explain this and say, why was B there before A? And here we're just essentially looking at, for SARS-CoV-2, that progression of mutations over time, which is something that we would see very early in the pandemic as well. And you can see here that sort of linear relationship. So there's a really good, strong clock-like signal, so that looking at samples, we can then estimate the age of the viral pandemic. And if we look at viruses from lineage A and lineage B, and we count the number of substitutions that separate them from the root, with time here, that's early 2020 on the x-axis, and number of substitutions away from the root on the y, you can see that lineage B, which is shown in turquoise, is just a couple of mutations farther from the root as lineage A over time. And in fact, along every step of the journey, B seems to be a little more diverged than lineage A. Or as the molecular clock would say, it looked like lineage B had a head start evolving and accumulating mutations, substitutions. So we can ask the question, lineage B looks like it is evolving more quickly, but is lineage A the ancestor of all of SARS-CoV-2, which you would think it should be because it looks more like the bat virus. So we went and basically performed phylogenetic molecular clock analysis on that tree. And here I'm going to just show you a bit of the results of that, which is basically a table showing what proportion of the time we inferred the ancestor to be lineage B. That's the percentage under that, what we call the unconstrained analysis, or how many times we thought the ancestor was lineage A, or we inferred the ancestor to be lineage A. Now you can don't bother looking at that part that says no market. That's when we threw out all the market data, which would just sort of lower our power to do anything here. But if you just look at that unconstrained sort of the basic analysis, you can see that the probability that the root should be at lineage B is around 80.85. And the probability that should be at lineage A is only 1.68. And in order to sort of describe that in a single number, you just take one probability and divide it by the other. And that's a fancy Bayesian process known as a Bayes factor. And that gives you your ratio, which here we say, well, it's about 48 times more likely that the root of the tree is B and not A. So the molecular clock doesn't like lineage A as the root. But we know that the bat viruses favor lineage A. So then we said, how can we incorporate all of the information about those bat viruses in? So we run across the entire SARS-CoV-2 genome and all of its most closely related bat viruses. And we found which bat viruses were most closely related along every segment of that genome and basically cobbled together sort of the hypothetical bat virus ancestor, which we here call the recombinant common ancestor, or RECCA. That's an acronym that you only need to remember for one more slide, I promise. So it's not important. You can forget it. 2 slides, forget what the RECCA is. But for now, know that it is the hypothetical, most closely related bat virus ancestor that we could infer. And if you root the SARS-CoV-2 phylogeny with that bat virus ancestor, the RECCA, you can see that these probabilities flip. And now, if you say, well, this is what we know the bat virus looked like, now the molecular clock wants to root it at lineage A, and you take 77.28 divided by 8.18, and you get a probability ratio of about 9.4. Saying now, if you think about the bats, it flips. So we have one situation where the ancestor is supported by the clock in humans, but it disagrees with the ancestor in bats. So why is the ancestor not supported by the molecular clock, or the presumed bat ancestor? And the answer would be that what if lineage B started transmitting in humans before lineage A got, or 2 introduction? One of lineage B, and then maybe a couple weeks later, lineage A starts to transmit. Which sounds ridiculous, because SARS-CoV-2, the COVID-19 pandemic, pandemics happen maybe once in a generation. A pandemic, the scope of COVID-19, hasn't happened since the 1918 Spanish Invasion. So how am I saying something that is literally a once-in-a-lifetime event happened twice in a very short succession? Well, and that's really unlikely, because you need everything to go wrong to get a pandemic the size of COVID-19. I would say you would need 9 misfortunes to get 1 zoonotic. You need to have a bat virus that is successfully transmitting in bats, has to evolve ACE2 binding, which is how it can bind to human cells, has to evolve what we call furin cleavage, so it can efficiently replicate in humans. You need to be able to infect a secondary host, like raccoon dogs or palm civets. It needs to be able to transmit in that secondary host. Then you need to be able to get one of those secondary hosts in very close proximity to humans. You need to be able to make the jump into humans. You need that virus then coincidentally to be able to replicate and transmit in humans. And only then, when all 9 of those conditions are met, can you get 1 zoonosis. And I'm saying we got 2.
But that will never happen. 3 misfortunes, that's possible. 7 misfortunes, there's an outside chance. But 9 misfortunes, I'd like to see that.
All right. I admit, improbable. You give up. Times like these, when you're despondent, you have to turn to a hero. Saint. The patron saint of all zoonoses. Who is that? George Clooney. Hey, George. What do you got for us?
Right. Who are you looking for? Tenzing Norgay. Tenzing Norgay.
Who's Tenzing Norgay? Patient Zero? Did he sell animals at the Huanan seafood market?
I doubt it. Tenzing Norgay was the Sherpa that helped Edmund Hillary climb Mount Everest.
Good. Unless. See, humans have lived in the shadow of Mount Everest for over 10,000 years. In all that time, not a single human being ever reached the summit of Mount Everest. Until one day, May 29th, 1953, two men reached Sir Edmund Hillary and Zenor. And they did it together because once they had the route, the gear, and the will to get up that mountain, the idea of one person doing it wasn't separate from two people. Probability of a second person making it up is conditional on the first person reaching the top. And then after the two of them did it, more and more people make the mountain. So in fact, the same is true of zoonosis. If you already have a bat virus that can bind to human cells and replicate in human cells with furin cleavage, and it can get into a secondary host, and you know it can transmit in that secondary host, and you know that that secondary host is living in close exposure to humans in a wet market, and you know that that virus is able to jump into humans, and you know that it can replicate and transmit in humans, and you know that that virus is already capable of causing the COVID-19 pandemic, to get a second introduction of a zoonotic virus capable of causing another pandemic, the only thing you need to do is maintain that exposure to humans of those secondary hosts in the horn. As long as you have that contact in place, you're going to keep getting zoonotic spillover and keep introducing. Now, if that's true, then lineage A should have been found at the market just like lineage B. Our hypothesis. Two spillovers at the market, both lineage A and lineage B should have both been at the market. Well, in fact, it was there the whole time. In 2023, the Chinese CDC published their paper on the metagenomic diversity of environmentally sampled viruses found at the Huanan Sea. And they showed that in the southwest corner of the market, right next to where all of the animals were being sold, there was a pair of gloves sampled on January 1st, 2020. And on those gloves was a lineage A virus. Lineage A was at the market the whole time, and the Chinese CDC didn't tell us, revealing that this virus likely started in bats, jumped into some still yet unknown intermediate host, and somewhere at the Huanan seafood market jumped into humans twice. Lineage B sparking the pandemic, and shortly thereafter, lineage A resulting in multiple zoonotic. Still trying to win the propaganda war here, but you've seen from our government, it's a slog. But with that, I just want to say thank you for coming today and for listening, and I hope you learned something about viral genomic epidemiology, and I'm happy to answer any questions.
Great talk. Thanks, Joel. Yeah, any questions from folks? Feel free to either unmute, feel free to type in the chat.
I'm just wondering, if lineage B was the first one to like spark and cause the first like cases in humans, how come that one is named like lineage B and then the second, like I guess wave, is lineage A? Like, does it not go in like in order in correlation to like alphabetical and like timeline?
Yeah, so it had to do with we started naming them after it was pretty diverse. And in fact, the Chinese named them S and N, I believe. And some group in Washington named them A and B, and they named it A because it was more similar to the bat virus. They said if we start with the bat viruses, this one looks like it should be the first lineage, and B should be. So that's how they got there. But B is the one that sort of stuck around, and that's how we have all the variants come from lineage. Alpha, beta, gamma. They're all lineage.
Yeah, and I posted in the chat. I don't know if Joel, if you want to talk about the Pango designation GitHub repo that is a
Oh,
very
yeah. So,
bureaucratic way that each lineage gets designated now.
yeah, lineages are, it's a very human process to sort of which lineages get special names and identifications. But it's also based sort of on the tree, and it's really gotten, it's something that I honestly haven't followed for years because now it's a very, it doesn't seem very relevant uh, since uh, since the Omicron subvariant waves kept knocking us down one after the other. But yeah, you should follow that link in there for a little more computation.
Any other questions, folks? Feel free. You got a world expert.
I was wondering, like, so you know how some of the hypotheses are like a little frivolous, right? Especially the one about the CIA. I was wondering if you've ever encountered anybody, because they're not specialists in epidemiology, like just like common folk who have like general knowledge, like have they ever tried to like discredit your work, or have they just like not believed like anything you said because they've fallen victim to like the whole Operation Infection?
Yeah, I mean the CIA one sort of fallen out of favor, although you will still sort of see that. But also it's
Is there
if
an
every
NIH
sort of
director?
new what?
Is there
Oh
an
yeah, I mean
NIH director
our
we may
current
have?
our current director of the NIH is very skeptical of our work and buys into these conspiracy theories. And in fact has threatened to defund pretty much the entire NIH and university research system based on the idea that the NIH had a hand in developing. And just 2 weeks ago, when he said this at an address to other scientists at the NIH, a bunch of them just got up and walked out on. So, what do they say? The call is coming from inside the house. That's where we are right now. But no, also I gave a talk to some researchers recently at another University of California institute, and in the question and answer round, I gave a much longer, more detailed version of this talk, and someone was definitely asking me questions that let me know that they'd been drinking the lab leak Kool-Aid. They knew a lot of probabilities for a lot of strange calculations that you got to be pretty deep in. So yeah, it's out there, and I don't know if it's more prevalent now than it ever was. I mean, it made it to the evening news in 1984, so like in some sense, it's worse now than it's ever been, and in another sense, it's always been bad. It's just bad in different ways. But to have it this deep into the U.S. government is really surprising.
So I have a question for you, Joel. So if I was a high school student who's thinking about what majors to pursue and what I want to study in college, what led you down this career trajectory?
Yeah. So I've been fascinated by viruses. I think since I was like 14 or so. I remember around then there was an outbreak of Ebola um, in then the DRC, and I remember reading an article about it and just being absolutely fascinated. And then in short succession, the book The Hot Zone came out, and then the movie Outbreak, which is not a great um, cinematic achievement, but it definitely struck a chord. 12 Monkeys is much better, far more science fiction, but really sort of the idea of like, oh yeah, you have to time travel in order to figure out what the ancestor of a virus looks like, because apparently they had forgotten about phylogenetics. But it's still, that type of fascination really got me there. And I sort of debated between like, following up with computer science, but my passion really was sparked by biology and sort of the contradictions and sort of paradoxes within it. And then when I discovered the molecular clock, I was smitten. And that didn't happen until my freshman year, but there are a whole lot of ways to go about it. If something really interests you, And it's not even something you need to be aware of now, but sort of go and explore what's there and just hold on to those early passions, because they do make life easier if you happen to find yourself in some career that sort of links in really, really care about. That's not to say it has to be that way, but it's a lot easier. So that's mine. And now the course I teach here is the intersection between movies and viruses. I teach a class on the movie Contagion. We walk through a very realistic, scientifically accurate depiction of a pandemic. Sort of learn more, pretty much what I just shared with you. There's a lot of find things that overlap and that make you happy. I don't know. Does that answer the question?
That was perfect. Absolutely.
Yeah.
Awesome. Any last questions before we call it? We're at like 2 minutes over, but I just want to give folks an opportunity.
Absolutely.
No? Awesome. Let's give our speaker another round of applause. Thanks so much, Joel. That was a great talk. I love all the movie references. Your game is on point with these like Zoom lectures. This is awesome. Awesome. So thanks so much, folks. So this is recorded, so I'll post it afterwards just in case anyone wants to rewatch it or show it to your parents, or anyone that couldn't make it, they can watch it. But yeah, I will see you all next week. Thanks so much, folks. Bye, everybody.